Hello grade 11 students. Today I am going to discuss the second lesson of your science textbook, that is photosynthesis. As you know, this is a biology lesson. You are familiar with this process. First we will discuss the term photosynthesis. We can break it into two components. What is the meaning of photo? It usually refers to light or light energy. And when we say synthesis, that means there is something being produced. So here, food being produced in the presence of light. And this process is carried out by plants. That is why it becomes a biological process. So photosynthesis is the process of producing food in the presence of sunlight by green plants. With that introduction, we will see the topics that will be discussed under this lesson. Factors that affect photosynthesis. So to understand the process of photosynthesis, you will need to know the factors that affect photosynthesis. The products of photosynthesis. Although we say food, we should know what the food is and what the byproduct is. And finally, the importance of photosynthesis. Now, we will move on to the lesson. There is an assignment given to you. Identify the organisms in the picture. So here you can see it's a grass meadow with plants, tall trees there, and you can see the sun, and there are two main types of animals. You can see the cow and the bird. I will note down the animals and the plants. This is a bird, and here we can see a cow. So that is again an animal. And here we have the plant and the grass. We have the plant and also grass. So out of these four organisms, you all know the basic classification. Plant and grass both come under kingdom Plantae, and cow and the bird comes under kingdom Animalia. But they are all living organisms. State their modes of nutrition. So here they want you to state their mode of nutrition. So to do that, what is nutrition? Nutrition is a process by which all these organisms obtain energy and material for the maintenance of their life. That is to grow, to do their day-to-day activities, to carry out their metabolic processes. For all these organisms need energy and materials. So to obtain that material and energy, they have different modes of nutrition. So here, if we consider the modes of nutrition, both these plants as well as the grass, they produce their own food. So what do we call them? They are autotrophic organisms. So these organisms show autotrophic nutrition. Then if we take the animals, bird and the cow, and there can be other microorganisms which are not visible to our naked eye as well. So these two, they can't produce their own food. So what do? They feed on other living organisms. So they obtain their food and energy from other organisms. So what are they known as? They are heterotrophic organisms, or they show heterotrophic nutrition. These are all very familiar to you. Now let me summarize what nutrition is and the modes of nutrition. So when we say nutrition, it is the process by which organisms obtain energy and materials for the existence of their life. And under this, we have already discussed there are two types. One is the autotrophic nutrition and the other one is the heterotrophic nutrition. So, as you know students, autotrophic nutrition is of two types. If the organism uses sunlight for production of food, then we call it photoautotrophic organism or photoautotrophism. So, under this we have photoautotrophism. But there are organisms, especially the bacteria. Most of the green plants carry out photosynthesis. So, they are photoautotrophic. But most of the bacteria, they use chemical reactions to produce their energy. Then they are known as chemoautotrophic organisms. So, that process is chemoautotrophism. So, these are the two main methods of autotrophic nutrition. There are different heterotrophic nutrition modes. You all know about the herbivores, carnivores and omnivores. But I am not going to go into detail there. In this lesson, we will focus on the photoautotrophic organism. Those are the green plants and how they carry out photosynthesis. So, we will discuss the process of photosynthesis in detail. And with that, I will move on to the lesson. Factors necessary for photosynthesis and its products. So, in that, we will first look at the factors that are needed for photosynthesis. Here you can see this is the sun. From the sun, you all know very well students, there is light energy and heat energy coming to the earth in different forms of radiation. So, there are different types of waves present in sunlight. But the plants absorb certain types of waves. So, here this is mainly the sunlight that is absorbed from the sun's energy. Energy is absorbed in the form of sunlight. So, that is why we call it as photosynthesis. Then it needs another factor that is absorbed by the roots. The roots normally have root hairs in order to increase the surface area so that they can efficiently absorb water. So here the root hairs absorb water that is present in soil. So here water is obtained from soil, the light energy from the sun. Then there is another factor that is necessary, that is in the gaseous state. Carbon dioxide that is present in the atmosphere. So here it is carbon dioxide is obtained from the atmosphere. Now to obtain this carbon dioxide, there are openings on the surface of the leaf called the stomata. You all are familiar with that structure. The stomata are usually covered with guard cells. And normally there is stomata present on the upper epidermis, that is the top surface of the leaf, as well as the bottom layer, that is the lower epidermis of the leaf. And usually there are more stomata in the lower epidermis of the leaf. And the carbon dioxide that goes in through the stomata diffuse into the leaf. So they just spread throughout the leaf through the intercellular spaces. So for this to happen, there needs to be stomata, enough amount of stomata, and carbon dioxide will diffuse into the leaf. When all these factors are present, photosynthesis doesn't take place. Why? That has to happen in a certain part of the leaf. As you know, there are parenchyma cells in the leaves. Two types of parenchyma, the palisade parenchyma and the spongy parenchyma. They are given a common name, the mesophyll cells. And these mesophyll cells or mesophyll parenchyma contain chloroplast. You know this organelle. And within the chloroplast, there is a colored pigment called the chlorophyll. So here, chlorophyll is a colored pigment present in chloroplast. And this photosynthesis process takes place in this chloroplast. So there are 4 factors. As you can see, sunlight, water from the soil, carbon dioxide from the atmosphere. These are all factors that are obtained externally. And out of these 3, water and carbon dioxide are raw materials because they will be used to produce the food in the presence of sunlight within the chloroplast that contains chlorophyll. And chlorophyll is present internally. And when the photosynthesis process takes place, there are 2 products produced. One is the glucose, that is the main product of photosynthesis, and the other one is oxygen gas. That is the byproduct of photosynthesis. So students, you have to remember the 4 factors and the raw materials, carbon dioxide and water, combine in the presence of sunlight within the chlorophyll to produce glucose and oxygen. So that process is known as photosynthesis. Utilizing the light energy, the cells containing chlorophyll in green plants synthesize food using carbon dioxide and water as raw materials. And this process is called photosynthesis. So here students, you have to look at this term. When we say green plants, the leaves of the green plants are mainly adapted to carry out photosynthesis. That is why they are green in color, and also they have all the required adaptations to carry out photosynthesis. But in young plants, you would have seen the stems of the plant being green in color. That means the stem, the young stem, also contains chlorophyll. So then they also can do photosynthesis. You would have seen the aquatic plants like Hydrilla and Vallisneria. Both their stem and the leaves are all green in color. So they will contain chlorophyll, they can do photosynthesis. Now we will see the water, that is a raw material necessary for photosynthesis. I said water is obtained from the soil, but it has to travel up to the leaf. Now we will see the way in which water from soil reaches the mesophyll cells of the leaves. Here you can see a structure of a plant is shown here, and water is being absorbed by root hairs. As I said before, this is to increase the surface area so that mostly water can be absorbed very efficiently. So then what happens is the water that enters into the root, it has to pass the cortex as well as the endodermis of the root. So here water from soil absorbed by root hair passes cortex and endodermis. So here students, you have to remember there is the epidermis as well as the endodermis. If you can remember, endodermis is present somewhere at the end of the cortex, just before the vascular bundle or the xylem and the phloem. So then the water enters the xylem tissue. That is the main transport tissue present to transport water. So here it goes into xylem of root. From there, it travels to the xylem of stem, then reaches the veins of the leaves. They also contain xylem. So, xylem of veins, and finally they reach the mesophyll cells. Then the mesophyll cells. And here students, you have to remember the process by which water is absorbed into the root hairs or absorbed by the root hairs and transported into the xylem of the root. That absorption process is known as osmosis. So here this process is called osmosis. We will not be discussing about osmosis in detail, but you have to remember this word. Osmosis is the process by which water is absorbed by root hair. So this is one raw material. I already explained how carbon dioxide enters into the plant leaf through the stomata and they diffuse into the mesophyll cells. The third factor that is obtained externally is sunlight. And plants have many adaptations to absorb sunlight very efficiently. So to do this, you have an assignment. Assignment, prepare a report on adaptations shown by plants to absorb light energy from sunlight efficiently for photosynthesis. I am sure students, you are very much familiar with all these adaptations. If you look at a plant and observe closely, you will see there are many adaptations in order to absorb sunlight efficiently. First, you all write them down and then listen to the discussion. So first, if we think of how a plant grows, the shoot system comes out of the soil and it grows usually upwards, but you have seen lot of plants growing towards the sunlight. And what do we call that process? That is phototrophism. You all are very familiar with that trophic movement. So normally plants grow straight and they try to grow tall so that they can get enough sunlight. But if the sun shines from a certain direction, the plant grows towards the sunlight. That is phototrophism. So that's one adaptation shown by plants. Plants grow straight and tall, and also they show phototrophism. If the plant stem is not strong enough, if they have a weak stem, then they can't grow straight or they can't bend towards the sunlight. Then what do? They grow around a substrate. For example, the climbers. And you have seen the epiphytes, certain plants that grow on a higher surface just to get enough sunlight. So there are climbers that grow on a higher surface or grow around a large plant, and the epiphytes that grow on a tall surface in order to obtain sunlight. And then if you look at the leaf structure, usually the leaves are at a right angle to the stem. It's always or mostly like this, or at least at an angle. The leaves don't grow this way. If they grow this way, they can get enough sunlight. And also you have seen they have a very large area. The leaf blade has a large surface area so that they can absorb the sunlight very efficiently. So the next one is the large surface area of leaf blade. And also you have seen different arrangements. Sometimes the leaves are opposite to each other. Some plants have leaves opposite and alternate. Two are like this, the other two are like that. So that all the sunlight can be absorbed by all the leaves efficiently. Then there are other leaf arrangements where there is a whorl arrangement, or there is a random arrangement of leaves so that some leaves are longer, some leaves have shorter stem, so that all the leaves get enough sunlight. So that is also an adaptation to absorb light energy efficiently. So the next one is leaf arrangement. So as you can see, students, there are many adaptations. If you think of the plant in more detail, if you go into more detail, or you can read a book, you can refer from reliable sources. you will see there are more adaptations that is shown by plants in order to absorb sunlight efficiently. With that, we will move on to the next part, factors that affect photosynthesis. We have already discussed the 4 factors: carbon dioxide, water, then we have the sunlight and chlorophyll. So, if we write down the factors, there are 4 factors that affect photosynthesis: carbon dioxide, from the atmosphere, water from soil, and these two are raw materials or reactants. Then we have the third factor, that is sunlight or light energy. All these 3 are obtained from the external environment. Then we have the fourth factor, chlorophyll, present in chloroplast. This is a factor that is present internally. So here it is a factor present internally. So as you can see, students, you have to remember the 4 factors. The factors that are obtained from the external environment, you should know from where they are obtained. Then you should know that chlorophyll is present internally. But if they ask you for the raw materials or the reactants of photosynthesis, it's only carbon dioxide and water, because those are the substances that will be used to produce food. Now we will see how chlorophyll is present in chloroplast by doing this activity. Observe chloroplast in plant leaves. This is a very simple activity. It needs an aquatic plant. So here I will need an aquatic plant. Aquatic plant, so for that we can use Vallisneria or Hydrilla. Then we need a light microscope. A light microscope, and we need a glass slide. So here we take a clean glass slide, take a small piece or a small part of the Hydrilla or Vallisneria plant, place it on the glass slide and observe that through the light microscope. So it's a very simple activity. The method: place a small section of the aquatic plant on a glass slide. Why do we need an aquatic plant? One thing, you can take them and place it on a glass slide, put a drop of water. Since they have the water environment, they will be alive, and also because of their nature, you can observe the chlorophyll or the chloroplast very clearly. Observe a small section of a Hydrilla or Vallisneria plant leaf under the microscope. Observe the way that chloroplast with chlorophyll move towards the direction of sunlight for photosynthesis. So here, students, you have to observe the chloroplast with chlorophyll. When we Place this slide under the light microscope and observe it through either low power or medium power. You can see the cells with the green color pigments in it. You cannot differentiate the chlorophyll or the chloroplast separately, but you can see the green color. If you move on to a higher power, then and if you place the microscope in a place where there is sunlight falling from a certain direction, then you can see that the chlorophyll containing chloroplast will move towards the direction of the sunlight. So this is a very simple activity. Now let us do this activity in the lab. Now we are going to do this activity to observe that chlorophyll is present in leaves. As you know, students, chlorophyll is the internal factor that is necessary for photosynthesis because it's already present in the leaves of green plants. For this observation, we need to use the aquatic plant hydrilla. So here you can see I have a small part of the hydrilla plant. From that, I have put it in water so that the plant is still alive, and I will take a small part of a few leaves from this hydrilla plant, and here I have a clean glass slide. On this, the lesser the leaves, it's better, so I'll put only a few leaves, and also to keep it hydrated and to provide enough water, I will put a few drops of water there. And as you know, without trapping air bubbles, we will have to close it with a clean cover slip. So here I have the clean cover slip and I have prepared the slide containing hydrilla plant. Now for us to observe it under the light microscope. I can place the slide on the stage and make sure there is enough light there. I will set it to low power and also move the slide around so that there is enough light falling on the leaf. Now when we observe it through the low power, you can see the green color chlorophyll or chloroplast present in the cells of the leaf. Here you can see the greenish color cells with the chloroplast in them. But this is not enough to observe it clearly because we will need a higher power to observe the movement of the chloroplast. So I will move it on to higher power. Now you can observe clearly the cells, that is the chloroplast present in the cell. Here you can only observe the chloroplast. But if I take this microscope to a place where there is strong direct sunlight, then since the plant is still alive, you can see the chloroplast moving towards the sunlight, towards the direction of the sunlight. But here, since it's inside the lab, we don't have enough direct sunlight, so you can only observe the chloroplast present in leaves. So students, now through this observation, you have seen that there is chloroplast in leaves, and that is the internal factor that is necessary for photosynthesis. Okay students, now you saw how cells containing chloroplast look like under the light microscope. We were not able to observe the movement of chloroplast with chlorophyll, but you were able to see the green color in the cells. So from that you can understand there is chloroplast that contains the colored pigment chlorophyll, and this is necessary for photosynthesis. And this is an internal factor that is necessary for photosynthesis. So, as we have already discussed, the green plants produce food using carbon dioxide and water in the presence of light within the chlorophyll that is present in chloroplast. Now, we will discuss the products of photosynthesis. Products of photosynthesis. There is a main product, that is the food that is going to be produced. And you are already familiar with this. The main product is glucose. And this you know is a monosaccharide. And the formula is also familiar to you. Glucose is C6H12O6. Now this glucose that is produced as the main product of photosynthesis is there in plant leaves. It's present in the mesophyll cells, but it cannot be stored in the mesophyll cells as glucose. So temporarily, glucose will be converted to starch and stored in the leaves. The glucose produced during photosynthesis will be temporarily stored in leaves as starch. And starch, you know, is a polysaccharide. And when the photosynthesis process takes place continuously, there is lot of glucose being produced. So this glucose is temporarily stored as starch, but it cannot remain in the leaves for a long time. So it has to be transported to the other parts of the plant. And that process is known as translocation. And in what form does translocation of food take place? It is translocated in the form of sucrose, and then we call it the phloem sap. And phloem is the transport tissue that translocates food in the form of sucrose. So food is transported into other tissues in the form of sucrose, that is a disaccharide present in phloem sap. These are all familiar to you. And the name of the process is translocation, and also it is transported through the phloem. And this is how the food that is produced, that is glucose, is converted to starch and then to sucrose and is translocated by the phloem. And when it is translocated to various parts, it again converts back to starch. Food transported to storing organs. Organs where the food is going to be stored are again stored as starch. And this is an example you have learnt before. Starch is present as the storage carbohydrate, and it is present in fruits, vegetables, yams, cereals, even lentils. You all know many different types of food that contain lot of starch. For example, potato, rice, different types of yams, lentils, fruits like banana, all these contain starch. So this is the main product. In addition to that, we discussed that oxygen is produced. And here that is the byproduct. So byproduct is oxygen. And this oxygen will be evolved into the atmosphere through the stomata again. So carbon dioxide comes in through the stomata It is used up by the plant leaf during photosynthesis. Oxygen is produced and oxygen is given out through the stomata. So there the plants, while producing food, they also clean the atmosphere. When we say clean, they help to maintain a balance between oxygen and carbon dioxide in the atmosphere. Reduce the carbon dioxide amount and increase the amount of oxygen. So here this is evolved through the stomata. So students, now you know the process of photosynthesis, the factors necessary and the products. We can put all of this into an equation form. We will discuss that. Photosynthesis can be expressed by a word equation as given below. So to write the word equation, we need to have the raw materials, carbon dioxide, and water. And for the process to take place, we need light energy, or you can write it as sunlight, and also there should be chlorophyll. That is the colored pigment. And the products are glucose and oxygen. Glucose is the main product, oxygen is the byproduct. So when they ask you to write the word equation, you have to write everything in words. Similarly, we can write the balanced chemical equation. So here for carbon dioxide, you all know it's CO2 and water H2O. Here we need to have these two, the light energy and chlorophyll. For this you don't need to give chemical formula, light energy and chlorophyll, and glucose is C6H12O6 and oxygen you all know is O2 gas. This is the chemical equation. It says balanced chemical equation. You all know how to balance chemical equations. There are 6 carbons in glucose in the product. So we need to have 6 carbons here. So 6 CO2. And if you look at the number of hydrogens, 12. So to get 12 hydrogens, again we need 6 water molecules. So the number of carbons, 6, hydrogen 12, and we have 6 into 2, 12 oxygen, plus again another 6 oxygen. So 18 oxygen atoms. Out of those, there are 6 oxygen atoms in glucose. So how many oxygen molecules will be evolved? There will be 6 O2 gas evolved. So this is the balanced chemical equation. 6 CO2, 6 H2O, light energy and chlorophyll to produce C6H12O6 glucose and evolve oxygen gas, that is 6O2. So these are the equations for photosynthesis. Now we will see what type of light is absorbed by plants. Extra knowledge: plants absorb red and blue light of sunlight during photosynthesis. We have already discussed how efficiently plants absorb sunlight. And you all know the sunlight, that is white light, that is in the visible region, it contains 7 colors. What are the colors? Violet, indigo, blue, green, yellow, orange, and red. Out of these, blue and red are the colors absorbed by plants. When we discuss waves, you will understand the difference between the different colors. They have different properties. So, out of those, blue and red that have different frequency and wavelength are absorbed by plants for photosynthesis. So, now that we know all these are the factors that are necessary for photosynthesis and what is produced during photosynthesis, we'll carry out some activities to understand these factors in more detail. So, the first activity, to identify starch produced during photosynthesis. Although glucose is produced, it's temporarily stored as starch in plant leaves. So, we can't identify or test for the glucose present, but we can test for the starch that is temporarily stored. To do this activity, we need all these materials. We need a beaker, a boiling tube, then we need a tripod with the mesh, a Bunsen burner, then we need water, we need ethyl alcohol, and we need leaves. So, this activity has many steps. First, we need to get a leaf that has been exposed to sunlight. So, you pluck a leaf from a plant that was present under direct sunlight. So, photosynthesis would have taken place and there will be starch present in leaves. So, here I will mention that exposed to sunlight. Then what we have to do is we have to put this leaf into a boiling tube, or we can directly use a beaker. Into the beaker we need to take water, put the leaves into water and boil it using the tripod mesh and the Bunsen burner. When we do this, when leaves are boiled in water, the cell wall and the cell membrane get damaged. Normally, you all know cell membrane is semi-permeable. It doesn't allow certain substances to come out or go into the cell. So, therefore, the cell membrane will not allow chlorophyll to come out of the cell. By boiling it in water, we remove this selective permeability. Or in other words, we can say we increase the permeability of the cells of the leaves. The permeability is increased by boiling the leaves in water. Then in the second step, we remove that leaf from the water, put it into a boiling tube. Into that we add ethyl alcohol enough to cover the leaves, and then we boil this using a water bath. That is very, very important. Why? Ethyl alcohol is highly flammable. Ethyl alcohol is highly inflammable. So, therefore, we have to use a water bath. And when we do this, you can see a change in the nature of the leaf, the color of the leaf and the solution. Then we remove the leaves from the boiling tube and wash it with water in order to remove the color as much as possible. After that, you will get a almost pale color transparent leaf. And then to that leaf, we will have to add iodine solution. So here we will also need iodine solution. You all know this is the reagent that is needed to test for the presence of starch. What is the color of iodine? It is a reddish brown color solution. And what is the color change you observe with starch? Iodine becomes dark purple or dark blue in color. So if we see that observation, that means there is starch present in the leaves. And therefore we can say photosynthesis has taken place. Now we will look at the method again. Here you can see the leaf being boiled in water. So there the leaf is inside the beak. No problem. But when we use alcohol or ethyl alcohol, we have to use a water bath. That is very, very important. So pluck a leaf from a plant which was in sunlight. Boil the leaf in water, that is to increase permeability. Then place it in the test tube with alcohol and boil it in a water bath. This is because it's highly inflammable and also it's very volatile. Quickly it can evaporate and it can catch fire. So to avoid that, we boil it using a water bath. And when we do that, the chlorophyll that is present inside the leaves will dissolve in alcohol. So this is to dissolve the chlorophyll. Then again we wash the leaf with water. So to that is again to remove the color, remaining color. And finally we test it with iodine. You know the color of iodine solution, reddish brown color. So here students, you have to remember the order of the steps and mainly you need to know why we carry out each step. Boil it in water to increase the permeability. Boil it in alcohol using a water bath because alcohol is highly inflammable, and by boiling it in alcohol we dissolve the chlorophyll. And then again we wash with water so that the remaining color will be washed off with water and also the alcohol that is present can be washed off with water. And finally, we test with iodine solution to see and confirm that there is starch present. So now that you have understood this activity, we will do the activity in the lab. Now we are going to test for the presence of starch in leaves. As you know students, starch is formed after photosynthesis takes place in the leaf. You know the main product of photosynthesis is glucose. And the glucose gets stored temporarily in the leaf in the form of starch. So if there is starch present in the leaf, that means photosynthesis has taken place. We have discussed this before. So now we will do the activity to test for the presence of starch. For that, I have taken a leaf, a shoe flower leaf. You can take any leaf like this, where you can easily observe the color change after we carry out the procedure. So, you know the first step, students? The leaf is not permeable, so we have to boil it in water. So, for that, I have a beaker containing water ready. Into this, I will have to put this leaf. I will put the leaf in. I have selected a leaf that can fit into this beaker, so that it is easy for us to boil it and take it out. So, I have the burner ready and now I have kept it to boil. You will have to observe the changes that are going to take place here. Initially, you saw that the leaf is green in color. So, students, now you can see when the water is boiling, you can see a change in the color of the leaf. Earlier it was green in color, now it is turning to a pale brown colored leaf. So, we will have to wait for some more time until it really becomes pale in color. And you know the reason for doing this first step. The leaf, the cells are not permeable. So, by boiling it, we increase the permeability of the cell by damaging the cell wall and the cell membrane. As you can see, students, the water is boiling and the leaf is getting boiled, so the permeability is increasing. Okay, students, now that the leaf is boiled enough, I will take it out of water. First, I will remove the leaf and place it on this. And also, I will need some more water because I am going to do the next step where I will need a water bath. So, to this, I will add more water. You all know, students, the next step we have to boil this leaf in alcohol, and this is the ethyl alcohol, which is highly flammable. It can vaporize quickly and catch fire quickly. Therefore, it is not correct to hold this tube to the direct flame. Therefore, I have to use this water bath. So, first I will transfer the leaf into the boiling tube and I will add enough alcohol to cover this leaf so that I don't put excess alcohol. I can prevent vaporization and alcohol catching flame. I will add some alcohol enough to cover the leaf. Here, if I add a small amount, that should be sufficient. Now, I will make sure that the leaf is completely immersed in alcohol. You can see I have put sufficient alcohol to boil the leaf. Here, we will place this inside the water bath. You all know the reason for this step, students. When we boil the leaf in alcohol, now that the permeability of the cells have increased, the chlorophyll will come out of the leaf and dissolve in alcohol. You can see the color change there. The alcohol solution is turning green in color. Earlier, when we boiled the leaf in water, there was no color change. The color of water remained the same. It was colorless. But here, alcohol is turning green in color. That means the chlorophyll is dissolving in alcohol. Okay, students, I think that is more than enough boiling in alcohol. The alcohol has become dark green in color. So most probably all the chlorophyll or most of the chlorophyll will be dissolved in alcohol. So I will remove it from the water bath. I have to place it, let it to cool a little bit. What is the next step? We have to wash this leaf in water. So for that again, I will take a little amount of water into the beaker and I will transfer this leaf. I can take it using the glass rod. Here you can see the alcohol is completely green in color. That means the chlorophyll has dissolved in alcohol. Now I make sure that the leaf is washed completely. Now I can touch it with my hand, no problem. So I will keep the glass rod aside and make sure that it has dissolved and got washed completely. Here you can see we have to make sure that the leaf does not get damaged. I will place it back in the petri dish. So now the leaf is ready for testing. So to see the color change clearly, I will transfer the leaf onto this piece of sponge. So now you can see the color of the leaf clearly and you can see this region where the color is very light. That means most of the chlorophyll has been removed from that region. Now using the glass tube, I will put a small amount of iodine onto the leaf. As we put it, unlike the starch test where you see the immediate color change, here you will not get the immediate color change. We will leave it for some time, then you will be able to see the color change. I will add a little bit more iodine to the other parts as well. We will have to leave it until the color change takes place. So now you can see the color change, students. In different parts of the leaf, you can see the dark bluish black color or the dark purple color that should form when starch and iodine are present. So this confirms that there is starch in the leaf. So that means photosynthesis has already taken place. So if you can remember, students, we took the leaf from a plant that was exposed to direct sunlight for few hours. And photosynthesis takes place in the presence of sunlight and therefore we can see the color change with iodine, the dark purple or dark bluish black color formed when there is starch. So with this we can confirm that photosynthesis has taken place. Okay, students. So now you saw what happens to the leaf when we do this activity. First we boil the leaf in water. Then what did you see? The green color turned to a lightly brown color. So initially the first observation, when boiled in water, leaf becomes brown. Then we boiled it in alcohol using water bath. Then what did you see? Alcohol became green, leaf became pale. This was the next observation. After that, we washed the leaves in water. Then you got an almost pale color, transparent leaf. After washed with water, pale, transparent leaf is obtained. After that, what did we do? We tested with iodine solution. When few drops of iodine solution was put on the leaf, it became purple or dark blue in color. So, color change with iodine solution. Purple or dark blue. So those were the observations. As I told you all students, you all should know the steps. You should know why we carry out each step, and you should know the change that takes place after each step. So if we get the purple or dark blue color, then we can confirm that there is starch present. So the conclusion, there is starch present in the leaf. What does that confirm? That photosynthesis has been occurred, or starch has been produced during photosynthesis. So here we can say the glucose, this is more better, glucose produced during photosynthesis is present as starch. So therefore we can confirm photosynthesis has taken place. Photosynthesis has taken place. So that is the first activity to test for the presence of starch, which is a temporary storage substance in leaves. Glucose is produced and temporarily stored as starch. Now we will move on to the next activity. Testing of factors required for photosynthesis. You all know there are 4 factors, and out of that, here we will show that light energy is required for photosynthesis. For that, we need to have a plant, usually a potted plant, a small plant, which can be moved to the lab and back to the garden to expose it to sunlight like that. So something that can be carried around easily. Then we need this black color polythene or black color tape. To have a control experiment, we need to have a transparent tape. These are to show that light energy is needed, but to confirm whether photosynthesis took place or not, we have to carry out the activity that we discussed before. So for that, we need water, alcohol or ethyl alcohol. Then we need a beaker to use as a water bath. We need boiling tubes. Tripod and wire mesh. And a Bunsen burner. And also we need to have iodine solution. That is to test for the presence of starch. So this is again an activity where you need to have a potted plant, and in that potted plant you have to select 2 leaves that are similar to each other. So if you select leaves that are at the same level and opposite to each other, most probably they will be of the same size and same maturity. So we select 2 such leaves and then we cover one part of the leaf, like say the middle part of the leaf on the top surface and the lower surface, above and the lower surface, we put this black color tape so that light does not penetrate into the leaf. To have a control experiment, to show that it is only light that is the factor that is being tested, we use a similar size transparent tape, and the same way we paste the transparent tape on the top and the bottom surface of the second leaf. So the 2 leaves are ready. But before we start the activity, there is an important thing. Now we are going to test whether light energy is required for photosynthesis. And how do we check whether photosynthesis has taken place? By testing for the presence of starch. You all know every day in the presence of light, plants do photosynthesis. So if you randomly take a plant, then the plant would have already done photosynthesis and there will be starch present in almost all the leaves. So we will not get the correct observation. So before we start the activity, actually 2 days before we start the activity, that is 48 hours before the activity, we need to take this potted plant and place it in the dark. You can keep it in a cupboard, close it inside a cupboard, or you can completely cover it with a black color, large black color bag, and you have to make sure that there is no sunlight falling on this plant for 48 hours. But we have to make sure that we give enough water. So you have to water the plant, then keep it in the dark for 48 hours. So that is the first step you have to do. We will discuss the method. So here you can see, select 2 leaves almost similar to each other and they are labeled as leaves A and B. And also the plant kept in the dark for 48 hours. This is very, very important. And this is to remove the starch produced before. So by doing this, we confirm or we make sure that there is no starch present in leaves prior to the activity. Then we cover the two leaves, one with black polythene, that is leaf A, and the other one with colorless or transparent polythene, that is leaf B, in order to make sure that we provide all the similar factors. Cover a part of leaf A with black polythene and part of leaf B with colorless polythene. Here I will also mention as transparent polythene, so that enough sunlight falls on leaf B, whereas there is no sunlight falling on this part of leaf A. Here again in leaf A, the other two sides, the tip and the base of the leaf will get sunlight. So that means we can make sure that the leaf is alive and also it can do photosynthesis. After doing this, we place this pot in direct sunlight for about 3 to 5 hours. Keep it under sunlight for 3 to 5 hours. And once that is done, we bring the plant back to the lab, pluck the two leaves. We make sure that we have them separately and we know which is leaf A and what is leaf B. Then we do the preparation of the leaf. Boiling it in water, boiling it in alcohol using a water bath, then washing with water, and finally we test with iodine solution to see whether there is starch. So that you have understood the activity now, students, we will carry out this activity in the lab. As I explained to you all, we are going to test whether light is necessary for photosynthesis. So for that, we have a potted plant. I kept this in the dark for 48 hours. So I took it out of the dark now and I am going to identify two leaves that are similar in nature, so that we can cover one leaf with black color paper and the other leaf with transparent polythene paper. So in order to do that easily, I have already taken two bags, sealable bags, so that it's easy to cover the leaves. One is in black color polythene, covered with black color polythene, and the other one is transparent. So then we provide identical environment except the factor sunlight. Now in this plant, if we look at the leaves, you can see this leaf on this side and the other leaf, these two leaves are all similar in size. So I will take two leaves that are opposite to each other. And then to one leaf, I will cover it with this black color polythene bag, but it is a sealable bag, so it's easy for me to cover it completely. In this activity, students, you can also cover part of the leaf with black color tape. You have the black colored tape, so you can paste it around the leaf. You can select an area and paste it as well. Otherwise, we can cover the full leaf with black bag. So with this other polythene bag, I will be covering an identical, we can't really say identical, a leaf similar to the first one that was covered with black polythene. So here, so now students, you can see the two leaves covered, one with transparent polythene bag and the other one with black color polythene bag. So there is no sunlight for this leaf and there is sunlight falling on this leaf. Apart from that, all the factors are same. Now I am going to take this pot and place it in a place where there is enough direct sunlight. And I'll leave it there for about 2 to 3 hours. Then we will do the test for starch. So we prepared two leaves, one covered with black color polythene, the other one covered with transparent polythene sealable bags. And then I place the plants in direct sunlight for about 3 hours. After that, I have prepared the leaf for y'all so that it's ready to test for the presence of starch. Y'all know the steps of this procedure. We have to boil the leaf in water first to make it more permeable. Then we boil it in alcohol using a water bath so that the chlorophyll will dissolve in alcohol. And since alcohol is highly flammable, we have to use the water bath. After that, when the alcohol solution becomes green in color, we stop boiling it and then we remove the leaf and wash it in water. Wash it well in water. After that, I have the two leaves here. Now, this is the leaf that was covered in transparent polythene. And this is the leaf that was covered with black color polythene. Now, to both the leaves, I will put iodine. If starch is present, then you will see a color change. You know the color change. It has to become dark purple or dark blue. If there is no starch, then there will be no color change. And the presence of starch indicates that photosynthesis has taken place. So, I put iodine to this leaf. I put enough iodine, and the same way I'll put iodine to the other leaf. You can already see the color change taking place. So, here you can see, students, the leaf that was covered with black polythene, the leaf that was covered with transparent polythene. And you can see that here there is no color change. The purple or dark blue color has not appeared. But here you can see there is the appearance of dark blue or purple color. That means in this leaf there has been photosynthesis, and this was covered with transparent polythene. So, light is needed for photosynthesis. The second leaf was covered with black polythene, no light, no photosynthesis. Therefore, you don't see a color change that has to be there if starch is present. So, from this activity, we can confirm what? That light is needed for photosynthesis. Okay, students. Now that you saw the activity in the lab, you saw what happens to the leaf. Because I could not put the tape, I used a black color polythene bag to cover one leaf completely and a transparent polythene bag to cover the second leaf completely. But if we have done it using tapes, then you would have seen that there was no color change in this, but in my activity, there was no color change for leaf A, but there was color change in leaf B. So, the observation will be like this. If I write down the observation, no color change is observed in the part of leaf A covered with black tape, but the other leaf had or showed color change. leaf B shows color change. What is the color change? The reddish brown color. Iodine turns purple or dark blue. So here what happens? There is no sunlight falling in this area because it was covered with black color tape. And there was no photosynthesis, so there is no starch and there is no color change. But the rest of the leaf and leaf B, there was enough sunlight falling on the leaf. Although leaf B was covered, it was covered with transparent tape. So sunlight can penetrate through that tape. So there was photosynthesis and there was starch. That is why it gave color. So from this, we can conclude that light energy is needed for photosynthesis. So that is the conclusion of the activity. Light energy is required for photosynthesis. So with that, we will move on to the next activity. Experiment to show carbon dioxide is required for photosynthesis. These are the materials that are necessary. We need a potted plant. What do you need to do for the potted plant? We need to keep the potted plant in the dark for 48 hours. We need to provide enough water and then keep it in the dark for 48 hours. Why? All the starch that has been produced before should be translocated to the other parts, and there should be no starch present in the leaf. Then we need a beaker. A beaker, we need boiling tube, the tripod, we need the Bunsen burner, we need water for the test for starch, then alcohol or ethyl alcohol, and here we need 2 polythene bags. Of the same size. And this is an important chemical substance. We need potassium hydroxide, that is KOH, and also we need iodine solution. So those are the substances we need. So first we keep the plant in the dark for 48 hours to make sure there is no starch present in the leaves. Then we select 2 similar leaves, almost the same height and opposite to each other, means they will be of the same maturity and size. We have 2 polythene bags so that light can penetrate through it. Into 1 polythene bag, we take potassium hydroxide. into the second polythene bag, we take the same volume of water. So if you take 10 milliliters of KOH, we have to take 10 milliliters of water. That is again to control the experiment. Same volume of solutions. And then insert one leaf into the bag containing KOH, seal it or tie it with a thread. The second leaf into the bag containing water. Again, seal it if it is a sealable bag, or we can tie it using a twine. And then we keep the potted plant under direct sunlight for about 3 to 5 hours. Then bring the plant back to the lab, pluck the 2 leaves and prepare the leaves. Before we prepare the leaf, we have to make sure what is the leaf that had been in KOH and the other leaf that had been inside the bag with water. And then prepare both the leaves separately. Boil in water, boil in alcohol using a water bath, wash it with water, and then we test it with iodine to see whether there is starch. Let me go through the method again. The method, here you can see the 2 leaves that have been selected. Leaf C, the bag has water in it, and the bag around leaf D has KOH solution in it. And here students, you have to know why we used KOH solution. I will discuss that after we do the activity. Select 2 similar leaves of the above plant, that is C and D. 2 similar leaves. Add potassium hydroxide KOH and water to transparent polythene bags. This is to make sure light goes in. Then insert leaf D into the polythene bag with KOH and leaf C into the bag with water and make them airtight. We have to make sure there is no air coming in or going out of the bag. Airtight, so that more carbon dioxide does not come in. Place the plant in sunlight for 3 to 5 hours. Then detach the leaves C and D and carry out starch test separately. So here, when we put KOH solution into the bag, it absorbs carbon dioxide from the air present in the bag. And once that happens, there is no additional carbon dioxide. To make sure that there is no carbon dioxide, we have to tie it airtightly. So those are 2 considerations in this activity. Now that you have understood the steps of the activity, we will do this activity in the lab. Okay students, you know the activity. We are going to test whether carbon dioxide is needed for photosynthesis. So the materials that we need, I have them here. A potted plant, it's a shoe flower plant, and then have the 2 solutions. One is water, that is labeled as C, you can see here, and KOH solution is labeled as solution D. The same time, I have 2 bags of equal size, they are transparent, so enough sunlight can penetrate to the leaves, and that is also labeled as C and D. In addition to that, I have written the liquids or the solution that we are going to use. Water in bag C and KOH in bag D. And to tie the bags to the leaves, I have the twines. So here from the plant, we will have to select 2 similar leaves. And this plant was kept in the dark for 48 hours. After that, I have brought it here, and now I'll take, you can see these 2 leaves are somewhat similar, size and opposite to each other, so the maturity is also the same. So I will select those 2 bags I'm selecting these two leaves. So I'll transfer the water into bag C and insert the leaf into the bag. And then we will have to tie it. Similarly, to the other leaf, I will add the KOH solution into the bag, insert the leaf, so here students, you can see one bag contains water, the other bag contains KOH, and this is leaf C, this is leaf D. Now we have to keep this potted plant in direct sunlight for about 3 to 4 hours. I will do that first. So now after keeping the plant in sunlight for about 4 hours, I brought the plant back to the lab. Then what do we do before we test for the appearance of a color change? We have to prepare the leaves. For that, we have to boil the leaves in water first. Then using a water bath, we boil the leaf in alcohol. Why do we use a water bath? Because alcohol is highly flammable. So to prevent it catching fire, we use a water bath. Then we wash the leaves in water, and now the leaves are ready for the final testing. So here I have the two leaves, the leaf C. In C we had water, and leaf D, inside bag D we had KOH solution. And I have the reagent that I am going to use, it's the iodine solution. So what we will do is we will put few drops of iodine onto both the leaves. I'll put it to leaf C first. You can observe the color change, and also to leaf D. It might take a little time. You can observe the two leaves. I will put a few more drops to both the leaves. So students, after I put few drops of iodine solutions to both the leaves, leaf C and leaf D, you can see the observation. You can see in leaf C there is a color change, appearance of purple or dark blue color. But in leaf D there is no color change. There is no purple color or dark blue color. So what can you infer from this? In leaf C there has been photosynthesis. So starch is produced and it gives the color change with iodine. But in leaf D there is no starch, that is why there is no color change. That means there has been no photosynthesis in leaf D. Now if you know the solutions, C was water and D was KOH. So water does not absorb carbon dioxide, therefore there is carbon dioxide for leaf C and photosynthesis takes place there. But for leaf D, there was no carbon dioxide. Why? KOH absorbed the carbon dioxide that was there inside that polythene bag. So there was no photosynthesis in leaf D. So from these two observations, we can confirm that carbon dioxide is necessary for photosynthesis. Okay students, now that you saw the observations of the activity, we will discuss why you get that observation and what we can conclude from the activity. So what were the observations? Leaf C that had water in the bag. What did you see? There was a color change. Color changes. What is the color that is seen or observed? The dark blue or purple color. To dark blue or purple. And what happens to leaf D? There is no color change when we test with iodine solution. So what is the reason? In leaf D, there was KOH, potassium hydroxide. And potassium hydroxide absorbs carbon dioxide present in air inside the bag. And also we make sure no air comes into the bag. So no air enters the bag later. So that means there was no carbon dioxide for leaf D. But for leaf C, there was enough carbon dioxide. So here leaf C had CO2, but leaf D no CO2. That is why we got this observation. So from this observation and the reason for that, we can confirm that carbon dioxide is necessary for photosynthesis. So here the conclusion is carbon dioxide is necessary for photosynthesis. Okay, students. With that, we will move on to the next activity. Experiment to show chlorophyll is required for photosynthesis. For this, we have to select a plant that has mosaic leaves. Mosaic leaf means here you can see leaves where there are yellow color regions. Green color regions that have chlorophyll and yellow color region that does not have chlorophyll. So the important thing is we need a mosaic leaf. For this, a good example is croton. So here we can use a croton leaf. Then we need to have A4 paper or any white paper on which you can sketch or actually draw the shape of the leaf and mark the regions that are yellow in color and that are green in color. Then to do the test for the presence of starch, we need water, we need alcohol, then we need to have a beaker to use as the water bath, boiling tube, the tripod, wire mesh and the Bunsen burner. So all these are necessary to test whether there is starch present in the leaf. So here we don't need to keep the plant in the dark for 48 hours. No need, because we are going to directly pluck a leaf, that is a mosaic leaf, for example a croton leaf, and then we draw the shape of the leaf by placing the leaf on a white paper. We draw the outline and then we mark the areas which are yellow and green in color. That is very important, because once we prepare the leaf for the starch test, you cannot differentiate the region that was yellow in color from the region that was green in color. Then we need water to boil the leaves. Once we boil the leaves in water, then we have to boil it in alcohol using a water bath. You all know the reason, because alcohol is highly inflammable. Then what do? We wash the leaf in water. Then you will get the transparent pale color leaf. Onto the leaf we add a few drops of iodine solution. Then you will get the observation. Okay students, so now that you know what we have to do, we will do this activity in the lab. Okay students, so you know the activity. We are going to demonstrate that chlorophyll, that is an internal factor needed for photosynthesis. For that we have to use a mosaic leaf. I have few examples of mosaic leaves here. You can see in this leaf these parts where you can see the yellow color regions. These are the places where there is no chlorophyll. Similarly, take this leaf. Here also in the middle and like a pattern, there is no chlorophyll. Another type of leaf is this one. This also you can see the green color regions and the off white or pinkish color regions that do not have the chlorophyll. So I am going to use this leaf for the activity today. What I have done is, already I have drawn the shape and of the leaf and I have marked the regions. Here you can see, if I place it here, it fits exactly with the leaf, and also you can see the regions that are shaded in the diagram is the region that has chlorophyll. So here you don't have to keep the plant in the dark for 48 hours. Directly I pluck this leaf from the plant. Now I have to prepare the leaf. You know the steps, students. Boil it in water, boil it in alcohol using a water bath, and then we have to test it for the presence of starch. So now I am going to do that procedure. So I have the leaf after preparing it for the test for starch. So to this we will add a few drops of iodine. Now I will add iodine to the full leaf so that you can observe the color change. We will have to wait for some time. Okay. Here you can observe, students, in the regions where there is the green color, that is chlorophyll is present, you can see the appearance of dark blue or purple color. But the regions where the leaf is completely not green in color, there is no color change. So from this observation, we can confirm that chlorophyll is necessary for photosynthesis. Okay students, so you saw the observations clearly. We used a mosaic leaf, drew the sketch of the leaf, and then we prepared the leaf for the test for starch. And when we tested, what did you see? Now here is a diagram of a certain leaf, not the same leaf I used for the activity. In this, you can see these are the green color regions. This region has chlorophyll and this region no chlorophyll. So the same way in this part when we do the test for starch, the region that has no chlorophyll does not give a color change. It remains the pale color. Whereas the region that had the chlorophyll gives a purple or dark blue color with iodine. So that is this region. Why does that happen? The region that was green in color does photosynthesis. So glucose is produced, it is temporarily stored as starch, and when we do the test for starch, we see the purple or dark blue color. But the region that is yellow in color does not have chlorophyll, so therefore it does not do photosynthesis. Therefore, there is no color change. So if we write down the observations, purple or dark blue color appears only in the green color part of the leaf. So from this we can conclude chlorophyll is necessary for photosynthesis. Okay, students. So now we did 3 activities. The activities were to show that carbon dioxide is necessary for photosynthesis, light is necessary for photosynthesis, and now chlorophyll is necessary for photosynthesis. You know that there are 4 factors. What is the 4th factor? That is water. Can we show that water is necessary for photosynthesis? Try to answer that question. Now we can't do this. Why? To show that water is necessary, from one plant we have to remove all water. But the plant will wilt and die if we remove all the water from the plant. So therefore, we cannot do this activity. An experiment cannot be designed in the laboratory to test the need of water for photosynthesis, because without water, the plant in the control experiment will die. So this is something that you already know and that you have to remember. So therefore, we can't do this activity in a normal manner. But scientists have found a way to show that water is needed. Scientists have shown the need of water for photosynthesis using water with 18,8 isotope. You all know what isotopes are. Atoms of the same element with different mass numbers. The common isotope is 16,8 oxygen. Here we use 18,8 oxygen. This is radioactive. Because of this radioactivity, by monitoring the radioactivity, scientists were able to show that water is necessary for photosynthesis. And they were able to do more research into how the process of photosynthesis takes place in a plant leaf. But that is not necessary for our subject content. So just remember that by using 18,8 oxygen, which is an isotope of oxygen, scientists were able to show that water is needed for photosynthesis. So far we have discussed different activities to show the factors needed for photosynthesis. Now we will move on to an activity where we show the product of photosynthesis. We have done the test to show that glucose is produced. We don't do the test for glucose, but we do the test for starch. What is the other product? The byproduct, oxygen. So the next activity is to show that oxygen is evolved during photosynthesis. Experiment to show oxygen is produced during photosynthesis. For this, we need these materials. We need a trough, a large container to take water. Then we can use a test tube or a boiling tube. Tube or test tube. Because here we don't have to heat the tube, it's just to collect the gas. So it can be any of the tubes. Then we need a glass funnel. And we need an aquatic plant. We need a plant, an aquatic plant. So as an aquatic plant, you are used to two common plants. One is hydrilla or vallisneria. So we can use one of these plants. What we do is we take water into a trough and into that we put this aquatic plant, and over the aquatic plant we place the glass funnel, and we make sure that the aquatic plant is contained within the funnel only. So we use only a small amount of the aquatic plant, enough to collect sufficient amount of gas that will be evolved during the activity. And on that we have to place this test tube. Before we do that, we fill the test tube with water, and without allowing any air bubbles to enter the test tube, we have to invert it over the funnel. That is very, very important. We have to make sure that there is no air bubbles or gas bubbles inside the water, that is inside the test tube. So it has to be completely filled with water. Then we place the setup in direct sunlight for about 3 to 5 hours. There you will see the observation. Okay, I will go through the method again. Add water into the trough. Then place hydrilla or vallisneria inside the funnel. Fill a boiling tube inside water trough and place it inverted on the funnel as in the diagram. Place the setup in sunlight. So in this diagram, you can see the trough, water, the aquatic plant and the light energy falling on it. And when that happens, you can see the gas bubbles coming up. and also the gas will get collected at the top of the test tube. So water will be displaced down. What is that process called? The downward displacement of water. So here we are collecting the gas using downward displacement of water. And from the equation, you all know it has to be oxygen gas. But we have to test and confirm that the gas evolved is oxygen. For that, what do? You all are familiar with that activity? We take a glowing splint or a glowing splinter and insert it into the gas that has been collected. You will see that the splinter will reignite and burn with a bright flame. So that confirms that the gas collected is oxygen. So this is a simple activity. Let us do this in the lab now. Okay, students. So now I have all the materials that needed to demonstrate that oxygen is evolved during photosynthesis. You all know glucose is the main product and oxygen is the byproduct. So I have a large beaker with water in it, and here I have an aquatic plant. You can take either Vallisneria or Hydrilla. I have here, this is Hydrilla plant. Then a funnel and additional water and also a test tube to collect the oxygen gas that will be produced during photosynthesis. So I will first prepare the setup. I will transfer the Hydrilla plants into the funnel. With that, if we can insert it into water, but I have to make sure all the plants are in water. Yes. So then I will need this test tube. There is little space. So what I will do is I'll add some more water, fill it up to the mouth. Before doing that, I have to increase the amount of water inside this so that there is no air or any form of gas inside the test tube. So if we fill the water above this level, and I will insert this, and then there is still gas bubbles, so I'll have to add more water. In this activity, students, we have to make sure that there is no air trapped inside the test tube. Now we can invert it inside water so that there will be no air bubbles inside the beaker. Now with this setup, in order for us to be, I will remove a little bit of water from this beaker so that we can easily carry it to a place where there is enough sunlight. So carefully holding it, I will remove a little amount of water. Now you can see, students, the setup is ready. The Hydrilla plant is inside the funnel and the test tube contains water only. There are no gas bubbles inside. Now we will have to place this setup in a place where there is enough sunlight or where there is direct sunlight. That is for about 2 to 3 hours. Then afterwards, when we observe, we can see the observations that need to be there. Okay, students. Here I have another setup that I prepared in the same manner, kept it in the sunlight for about 3 hours. Now here you can observe inside the funnel. If you look closely, you can see there are a lot of air bubbles. and also you can see that the water level has decreased a little bit, and on the sides of the test tube also you can see the air bubbles or the gas bubbles actually. So if we move it a little bit, you may be able to see the movement of the gas bubbles. You can see the gas bubbles coming up. So this is how the gas will evolve and get collected in the test tube. So you have to remember students, there will be observations. One is the evolving of the gas bubbles, you can observe that, and also the water level will decrease and the gas that you already know is oxygen will be collected on the surface of water. So that is above water. Water will be displaced and oxygen will be collected in it. If we still keep this setup in sunlight for some more time, there will be more gas collected. That is, there will be more oxygen collected in it. So now I will keep this in the sunlight and then we will observe it and test to check whether the gas collected is oxygen. Okay students, now I left this setup in the sunlight for few more hours. You can see the water level has decreased further and the oxygen gas is collected inside the test tube. So the next step, we will confirm that the gas evolved is oxygen. What do? We have to test it with a burning splint. So to do that, I will have to take out the test tube, making sure that the gas does not escape. So I will have to tilt it a little bit and then you can see there are more gas bubbles coming out. Okay students, now I will invert the test tube so that the air is on top. You can see it, and now I will insert the glowing splinter. You can see it glowing more. So that is because there is oxygen in it. So students, you saw that this was the normal splinter. Without, you couldn't see the glowing end. But when we insert it into the test tube with oxygen, you saw a better glow. If there was enough oxygen, a very large amount, you can see a flame. Because there was only a small amount of oxygen, you were able to see the glow only. So with that, we can confirm that the gas collected inside the test tube was oxygen. And we can also confirm that oxygen is evolved during photosynthesis. That is the byproduct of photosynthesis. Okay students, so you observe what happened in the setup. The setup that was prepared to show that oxygen is evolved in photosynthesis. So what did you see? You saw the gas bubbles evolving from the plant and moving upwards. So that was one observation. Gas bubbles evolve from the plant. Then what did you see? Gas was collected by downward displacement of water. Gas was collected by downward displacement of water. Then what did we do? We took the test tube containing oxygen gas, making sure that the gas does not escape the tube. And into that, we insert a glowing splinter. And then what do you observe? You see that the glowing splinter rekindles or reignites and burns. So that is the test to confirm that oxygen is evolved during photosynthesis. So here, insert a glowing splinter. So that is the test, a very simple test. And what do you see? It rekindles and burns with a flame, so that we can confirm it is oxygen gas. So the conclusion will be oxygen is produced during photosynthesis. Or you can say oxygen is evolved during photosynthesis. So now we have completed all the activities. So students, with that we have actually come to the almost the end of the lesson. Extra knowledge. Lavoisier is the first scientist who showed that oxygen diffuses from green plants in the presence of sunlight. So Lavoisier is a scientist who observed that during photosynthesis, he did not do research into photosynthesis, but he saw when there is sunlight, green plants. So those are the two important points. Green plants in the presence of sunlight diffuse oxygen or evolve oxygen. We need not go into more detail, but you all have to remember that he is the scientist that who first observed that oxygen is evolved by green plants in the presence of sunlight. Now we will move on to the importance of photosynthesis. Importance of photosynthesis. Light energy is converted to chemical energy during photosynthesis. So this is something very important. Light energy is converted to chemical energy. We get light energy from the sun, and the glucose produced is in the form of chemical energy. Plants produce food, and all organisms live on earth depend directly or indirectly on that food. At the beginning of the lesson, we discussed the different modes of nutrition. Autotrophic organisms produce their own food, but the heterotrophic organisms, if they are herbivores, they directly consume the plants. So they depend on the plants for food. If they are carnivorous animals, then they consume other animals which have already consumed plants. So either directly or indirectly, they depend for food on these plants. Photosynthesis cannot be done artificially. We cannot do artificial photosynthesis. Therefore, this process is carried out by green plants, is essential for the maintenance of life on earth. So this is one main significance, maintenance of life on Earth. The oxygen gas, which is required for the survival of aerobic organisms. So, oxygen gas, you know what aerobic organisms are. We have discussed cellular respiration to a certain extent. You all know inside the mitochondria, it is aerobic respiration that takes place. For this, the organism needs oxygen. So, that is obtained from this photosynthesis process. The oxygen gas which is required for the survival of aerobic organisms and combustion of materials is released mainly by photosynthesis. So, photosynthesis is the main process that releases oxygen to the atmosphere. Carbon dioxide that is accumulated due to respiration and combustion is removed from the environment by photosynthesis. So, here as you can see, carbon dioxide that is given out during respiration and combustion, and oxygen is needed for respiration and combustion. So, what happens is when photosynthesis takes place, carbon dioxide is removed, oxygen is added to the environment. Therefore, it helps to maintain oxygen and carbon dioxide balance in the atmosphere. So, it helps to maintain this balance. And finally, photosynthesis helps to maintain the carbon cycle. You all know elements go on or undergo changes in a cyclic manner. So, carbon dioxide is absorbed by plants, then it is converted to glucose, then to starch. It's stored as starch, consumed by animals, and then when these plants and animals die, again the organic material decompose and carbon dioxide is evolved back into atmosphere. So, that is just a simple cycle. In this, photosynthesis is a very important process. So, photosynthesis helps to maintain the carbon cycle. These are the importance of photosynthesis. Now, we will move on to the summary of the lesson. Summary. Light energy is converted into chemical energy by green plants during photosynthesis. So, you have to remember this energy change. Light energy to chemical energy. Light energy, water, carbon dioxide and chlorophyll are required for photosynthesis. And you all know students, chlorophyll is present internally. The other three factors are obtained from the external environment. And out of those, both carbon dioxide and water are the raw materials or the reactants of photosynthesis. The main product of photosynthesis is glucose and oxygen is produced as a byproduct. So, two products, main product glucose, byproduct oxygen. Photosynthesis can be expressed by a balanced equation as below. You all know to write the word equation as well as the balanced chemical equation. 6 CO2 gas, 6 H2O liquid. We didn't use these physical states before. That is to show in what form these substances are absorbed or used up and then produced. So, CO2 gas, 6 H2O liquid in the presence of light energy within the chlorophyll. Glucose, C6H12O6, that is a solid, and oxygen 6O2, that is gas. So, this is the balanced chemical equation. The global importance of photosynthesis is provision of food to all organisms directly or indirectly, maintenance of O2-CO2 balance in the atmosphere, and maintaining the carbon cycle. We have discussed these significance previously. So students, with that, I have completed the discussion on photosynthesis. As I always tell you, again read your textbook, watch the discussion again, try to understand everything completely, and then practice doing papers. So as a practice for you all, first I will discuss the questions in your textbook and then we will do some additional questions. The Unit 2 exercise. Underline the correct answer. You are familiar with these types of questions. First one, what is the main product of photosynthesis? I am sure you know the answer immediately. It is glucose. What is starch? Glucose is temporarily stored as starch in leaves and also it is stored in other parts of the plant. Sucrose is transported through the phloem in the form of phloem sap, that is translocation, and oxygen is the byproduct of photosynthesis. What is the tissue involved in transporting products in photosynthesis to storing organs? I already mentioned the answer. What is the tissue? It is the phloem tissue. So, xylem transports water. Phloem translocates food. And parenchyma is usually the storage tissue and collenchyma is for support. What is the food type that translocates to storing organs? That also we discussed. You all know it is sucrose. So, it's the first answer. Glucose is the main product and starch is the temporary storage and the storage carbohydrate. Cellulose is a component of the cell wall. We will move on to the next question. What is the gas emitted during photosynthesis? You all know the factors, carbon dioxide and water. Main product is glucose, byproduct is oxygen. So, answers carbon dioxide, nitrogen of course is present in very high amount in the atmosphere. So, it is oxygen and not carbon monoxide. Into which energy that solar energy is converted to during photosynthesis? You all know the energy conversion. Light energy is used to produce glucose. So, the conversion is from solar energy or light energy to chemical energy. Heat energy, light energy, chemical energy, potential energy. So, it has to be chemical energy. So, those are the answers. We will move on to the next question. Put right or wrong for the given statements. First one, color change with the starch test can be seen in a leaf after keeping it in dark for 48 hours. So, you know students why we keep the plant in the dark for 48 hours. To remove all the starch that is already present in a plant by doing photosynthesis. Photosynthesis, the main product is glucose. It is temporarily stored as starch. And when we keep the plant in the dark for 48 hours, all the starch will be translocated to other parts of the plant. So, here then we will not get a color change because the color change with iodine test is when there is starch present. So, here the statement is incorrect. Second one, leaf should be boiled in water to dissolve chlorophyll for the starch test. We do boil the leaf in water, but for what reason? To increase the permeability of the leaf. So, here it says to dissolve the chlorophyll. That is also incorrect. What do we do to dissolve the chlorophyll? We boil the leaf in alcohol using a water bath. So this statement is also incorrect. Then the third one. Photosynthesis takes place in leaves only. Is that correct? No. Why? Because in young plants, the stem is also green. So photosynthesis can take place in the leaf as well as the stem. And again, we have certain xerophytic plants. Plants that grow in very dry area where there is high loss of water. So to reduce the loss of water, the plant leaves are lost and the stem becomes fleshy and green in color. So there the stems are photosynthetic stems. So they do photosynthesis. So then photosynthesis takes place only in leaves. That is also incorrect. Then the fourth one. When leaves are boiled in water, the permeability of them increases. That is correct. We discussed it with the second statement. We boil the leaves in order to increase the permeability. Then the last one. Photosynthesis takes place only in green plants. That is also incorrect. Why? If we take an aquatic ecosystem, the main producers present in aquatic environment are green algae. They are photosynthetic algae. So they also do photosynthesis in addition to green plants. Mostly green plants do photosynthesis and also green algae do photosynthesis. So therefore, this statement is also incorrect. So students, now we have answered all the questions here. We have put a tick or wrong for the statements given. I will move on to the next question. When grass is covered for 3 days with a black colored polythene, it becomes yellow in color. Design an experiment to prove this phenomenon. State observations and conclusion. You all know why the grass turns brown in color. Grass is usually green. When light falls on it, they do photosynthesis. But when it is covered with black color polythene, there is no sunlight falling, so therefore there is no photosynthesis taking place. Therefore, they turn brown in color or yellow in color. So how can we show this using an experiment? We can select a certain area of grass, and in that we can take 2 small areas which are identical in size and that have almost the same amount of grass. And we can cover one area with black color polythene, another area with transparent polythene. What is the activity you remember now? The activity that we carry out to test that sunlight is necessary for photosynthesis. So here, design an experiment to prove this phenomenon. So what we can do is, we can select an area where there is grass and one area we can cover with black polythene. Another area we can cover with transparent polythene. Then you leave it like this for 3 to 4 days. And you can see that after 3 or 4 days, this part which has been covered with transparent polythene will still remain green in color. But this area which was covered with black polythene, the grass would be yellow in color. So that is the observation. What will be the conclusion? We can conclude and say, because there was no sunlight, the plant could not do photosynthesis. Therefore, the chlorophyll that was present in the leaves was destroyed or removed from the leaves. That is what we can conclude from this activity. So, this is a design of an experiment. So, here we can say select an area with grass, divide two identical areas. Then I will write it here. Cover, let's say A and B. A with black polythene, cover B with transparent polythene, then leave it exposed to sunlight for 3 to 4 days and then observe. Observe means we have to remove the polythene and observe. Then what will you observe? A, grass is yellow, and B, grass remains green. What is the conclusion? Because there is no sunlight, there is no chlorophyll. When there is no sunlight, plants lose chlorophyll. So that is the conclusion. So students, this is a simple design of an experiment. When you are doing it, you have to make sure that except sunlight, all the other factors are provided equally to two sets of plants, set A and set B. So with that, we have discussed all the questions in your textbook. Now I will discuss some additional questions. Additional question 1. Select the correct answer. Again, these are MCQ type questions, multiple choice type questions. One of the main raw materials needed for photosynthesis is main raw material: carbon dioxide, carbon monoxide, oxygen and starch. You know what the raw material is. It is carbon dioxide. We don't get or use carbon monoxide. Oxygen is the byproduct and starch is the temporary storage after photosynthesis. What is the main product of photosynthesis? That you know the answer. It is glucose. Hydrogen, glucose, sucrose and starch. So it's glucose. Next question. The tissue that transports the food produced by photosynthesis Cambium. Cambium is meristematic tissue. That is there for the lateral growth. That is the formation of wood in woody plants. Xylem transports water. Phloem translocates food. And collenchyma is a supportive tissue. So, the tissue that transports the food produced by photosynthesis is phloem. Solvent used to dissolve chlorophyll is. You can remember the activity, how we prepare the leaf for the test for starch. We boil it in water, that is to increase the permeability. We boil it in alcohol so that chlorophyll will dissolve. So, the solvent used to dissolve chlorophyll is alcohol, not water, salt solution or vinegar. It has to be alcohol. The next one. To what form is the solar energy converted by green plants during photosynthesis? You all know the energy conversion. From solar energy, we get chemical energy because glucose is produced. So, light, heat, molecular energy, chemical energy. What is the byproduct of photosynthesis? That again, you all know the products are glucose and oxygen. Glucose is the main product, oxygen is the byproduct. Water, oxygen, glucose and not sucrose. I'll move on to the next one. How is the food produced by photosynthesis translocated to other tissues? Glucose is produced, starch is temporarily stored and sucrose is translocated to the other parts. So, starch, glucose, sucrose. Fructose, this is also a monosaccharide that is present mainly in ripened fruits. What is not an adaptation of plants to absorb the sunlight efficiently for photosynthesis? Having broad leaf blades. This is an adaptation. You can remember, students, we discussed a few adaptations under your assignment. So, having broad leaf blades is an adaptation to absorb sunlight efficiently. The stem grows towards the sunlight, that is phototrophism, in order to obtain enough sunlight. The stomata open widely during the day. This is also needed for photosynthesis, but in order to get enough carbon dioxide into the leaf and to evolve the oxygen. So, the function of stomata is gas exchange. Having a very thin cuticle. Cuticle is the outermost layer that is present over the epidermis, upper and lower epidermis. When it is thin, it's easy for the sunlight to diffuse through the cuticle. Usually the cuticle is thin and also transparent. But in plants where they need to reduce the loss of water, there can be a thick cuticle. So, these are seen in plants which grow in very dry climate where there is lot of sunlight. Otherwise, the normal terrestrial plants have a thin cuticle. So, here as you can see, students, the first one having a broad leaf blade, the stem growing towards the sunlight and having a very thin cuticle are all adaptations to absorb sunlight efficiently. But this, the stomata open widely during the day, that is for efficient gas exchange. So, therefore, that will be the answer for this question. I'll move on to the next one. Additional question 2. A setup prepared to identify the presence of starch in plant leaves is given below. So, you know this activity as you look at the picture, boiling the leaf in water, and the next step, boiling the leaf in alcohol using a water bath. You are familiar with the steps. A leaf from a plant which was exposed to sunlight was plucked. First, it was boiled in water. Then the leaf was placed in the test tube with alcohol and was boiled using a water bath. The leaf was washed with water. The leaf was tested with a few drops of solution A and the color change was observed. So here they have given all the steps involved in this activity. We'll answer the questions related to the activity. Why do we need to boil the leaf in alcohol? I'm sure you know the answer to this question. In order to dissolve the chlorophyll and remove it. So here we can write the answer as to dissolve the chlorophyll. What will be the colors of the leaf and alcohol after boiling? Initially the leaf will be green color, alcohol is colorless. What did you see during the activity? Alcohol becomes green, the leaf becomes pale in color. So here this is after the boiling with alcohol. So leaf will be pale color, alcohol will be green in color. Why do you need to use a water bath when boiling with alcohol? Because alcohol is highly inflammable. To avoid it from catching fire, we boil it using a water bath. So here you have to say alcohol is highly inflammable. We use a water bath to avoid alcohol from catching fire. So we are actually preventing accidents. I will move on to the next question. What is solution A? What is the color of this solution? So in the last step, they said few drops of solution A was added and the color change was observed. You know what solution A is? It is iodine solution. So A is iodine solution. And what is the color of this solution? It is reddish brown color. What is the expected color change with solution A? The leaf will become purple or dark blue if there is starch. So that is why they say expected color change. So here we can say it turns purple or dark blue. If you want, you can write in brackets if starch is present. That is to confirm your answer and to make it more, give more information. What will be the conclusion of the activity if you observe the above color change? What will be the conclusion? Starch is present. Therefore, photosynthesis has taken place in the leaf. So here conclusion will be, since starch is present, photosynthesis has taken place in the leaf. So that is the answer. Next question. Additional question 3. An activity done to observe the evolving of oxygen gas during photosynthesis is shown below. You all are very familiar with this activity, students. We did this in the lab. Trough containing water, an aquatic plant, and a funnel is covering the plant. A boiling tube is inverted over it and a gas is collected at the top when there is enough light energy falling on it. Aquatic plant's hydrilla or vallisneria was placed inside a funnel. A boiling tube filled with water was placed inverted on the funnel as in the diagram. Then the setup was placed under direct sunlight. So this gives all the steps involved in this activity. Now let us move on to the questions. What are the observations of the above activity? You all can see the gas bubbles evolving from the plant and the gas getting collected with the downward displacement of water. So you can see the gas bubbles evolving and the water level decreasing. So those are the observations. Gas bubbles are evolved and you can see the water level goes down. How would you confirm that the gas evolved in the above activity is oxygen? You know the activity by using a glowing splinter. By using a glowing splinter. What will be the observation of the activity that you mentioned in part 2 above? That is when you insert a glowing splinter. It will rekindle or reignite and you will observe a flame. So here you can write the glowing splinter will rekindle and burn when inserted into the tube containing oxygen gas. So those are the answers for these questions. I will move on to the next question. Why can't you test the requirement of water for photosynthesis in the laboratory? I am sure you know the answer. In the control experiment, if we remove all the water from the plant will die. So therefore, we cannot do the activity. So that is the answer you have to write here. In order to do the control experiment water has to be removed completely from a plant. Then the plant will die. Therefore, we cannot do an activity to test for the requirement of water for photosynthesis. How do scientists demonstrate the requirement of water for photosynthesis? How do they demonstrate, or how have they demonstrated? By using 18,6 oxygen isotope. I told you all that is a radioactive isotope. By determining and monitoring the radioactivity, they were able to demonstrate that water is needed for photosynthesis. So here you can write the answer as by using 18,8 oxygen. This is an isotope of oxygen. If you write that, that is more than enough. Move on to the next question. Additional question 4. Fill in the blanks with suitable words. Green leaves of a plant can be considered as an industry based on the main process taking place in them. So you know what the process is? The process of synthesis that takes place is known as photosynthesis. The main product of this process is glucose. Raw materials are obtained by different methods. Dash is obtained from the atmosphere. What is obtained from the atmosphere? Carbon dioxide. So we can either write the word or I can use the formula. CO2 is obtained from the atmosphere and is obtained from the soil. What is obtained from the soil? Water. Water is obtained from the soil. Energy needed for the process is obtained in the form of sunlight. And is a colored pigment present within the plant leaves that is needed for this process. What is the colored pigment? It is chlorophyll. Where do you get this colored pigment? Inside the chloroplast. Then we have during the synthesis is obtained as a byproduct. Oxygen is obtained as a byproduct. This process takes place within the cells of the leaves. Within what cells? The parenchyma cells. They are given a special name. They are called mesophyll cells. And you know there are two types, the palisade and the spongy mesophills. So here we can write as mesophyll cells. Cells of the leaves. The exchange of gases occur through the, what is the opening present in leaves? Stomata. This process helps to maintain balance in the atmosphere. What balance? The balance between carbon dioxide and oxygen. So to maintain carbon dioxide and oxygen balance in the atmosphere. I will read the paragraph again. Green leaves of a plant can be considered as an industry based on the main process taking place in them. The process of synthesis that takes place is known as photosynthesis. The main product of this process is glucose. Raw materials are obtained by different methods. Carbon dioxide is obtained from the atmosphere and water is obtained from the soil. Energy needed for the process is obtained in the form of sunlight. And chlorophyll is a colored pigment present within the plant leaves that is needed for this process. During the synthesis, oxygen is obtained as a byproduct. This process takes place within the mesophyll cells of the leaves. The exchange of gases occur through the stomata. This process helps to maintain carbon dioxide and oxygen balance in the atmosphere. I'll move on to the next question. Additional question 5. Why are plant leaves green in color? Why are they green in color? Because they have the colored pigment chlorophyll inside chloroplast. So here we can say the green colored pigment chlorophyll is present in chloroplast. What is the function of chlorophyll present in plant leaves? Why do plant leaves need chlorophyll? To absorb sunlight and to carry out photosynthesis. So here you can say to absorb sunlight and to carry out photosynthesis. Next one. What is the substance obtained from the atmosphere for photosynthesis? What do you take from the atmosphere? The plants absorb carbon dioxide. Carbon dioxide. What is the material obtained from the soil for photosynthesis? It is water. Next question. Additional question 6. What is the part of the plant that is adapted mainly for photosynthesis? I discussed this with you all before. Leaves are the main parts of the plant that are adapted for photosynthesis. But in young plants, the stem also can carry out photosynthesis. And in xerophytic plants where they need to prevent loss of water, the photosynthetic stem can do photosynthesis. But here they ask you for the part that is adapted mainly for photosynthesis. Then we have to say the leaves. What are the factors necessary for photosynthesis? You know the factors: water, carbon dioxide, sunlight, or you can say light energy, and chlorophyll. explain how water from the soil is distributed throughout the leaves. You all know this process. The root hairs absorb water. Water travels through the cortex and the endodermis into the xylem of the root. Then from the xylem of the root to the xylem of the stem and to the xylem of the vein. And finally, they spread throughout the mesophyll cells. So that is what we have to explain here. So I will say water is absorbed from the soil by root hair. It enters the xylem of the root, passing the cortex and the endodermis. Travels through the xylem of the stem and then veins. Spreads into the mesophyll cells. So this is the path by which the water absorbed by the root hair comes to the mesophyll cells. Move on to the next question. Additional question 7. What is the main food produced during photosynthesis? You all know the answer. It is glucose. What is the gaseous byproduct of photosynthesis? It is oxygen. What happens to the gas you mentioned in part 2 above? Oxygen is evolved out through the stomata. It is released to the atmosphere through the stomata. Write the word equation for photosynthesis. You know this. Water, carbon dioxide, then on the arrow we have light energy, or you can write it as sunlight. And we need chlorophyll from this glucose and oxygen are produced. Then we have the next one. Write the balanced chemical equation for photosynthesis. You know that as well. 6 CO2 gas plus 6 H2O liquid. Students, you all know that the states gas, liquid and solid, they are all optional. you can write the equation without stating the physical states. Then we have light energy chlorophyll. These have to be written in words. Then we get glucose C6H12O6, that is a solid, and 6O2 gas. I am sure you wrote the equation correctly. We'll move on to the next question. Additional question 8. Draw a label diagram of the activity carried out to show that carbon dioxide is necessary for photosynthesis. I am sure you can remember the activity. So here we have to draw a label diagram. What did we do? We took a potted plant, kept it in the dark for 48 hours, and then we selected 2 similar leaves, and into one polythene bag, transparent polythene bag, we take water. Into another transparent polythene bag of the same size, we take the same volume of potassium hydroxide, insert one leaf into one bag, tie it with a twine, and the other leaf into the other bag, tie it with a twine. We have to make sure that air doesn't go into the polythene bag. Then we leave it in the sunlight for 3 to 5 hours. After that, we pluck the 2 leaves separately, prepare them for the test for starch, and then you will get the required observation. So here we only need to draw the label diagram. So here you will have to draw a potted plant with 2 similar leaves. Maybe this also has more leaves. You don't have to remove the rest of the leaves. And around this we tie a bag. In this we take water, a small amount of water. Around the second leaf we have another bag that has KOH, or you can write it as potassium hydroxide. Then what do? We leave this setup in the sunlight and then prepare the leaves and test it for the presence of starch. So since they have asked for the label diagram, if you show this, that is enough. If we want, here we can mention a potted plant kept in the dark for 48 hours before the activity. That is this. So here we have included all the necessary information. I will move on to the next one. Additional question 9. An experiment cannot be designed in the laboratory to test the need of water for photosynthesis. Why? As a control experiment, if we remove all the water from a plant, the plant will die. So therefore, we cannot do the activity. So we have to write the answer as a control experiment, if all the water is removed from a plant, it will die. So therefore, we can't do this activity. How did the scientists show that water is necessary for photosynthesis? We have answered this question before as well. I am sure you know the answer by using 18,8 isotope of oxygen. By using the 18,8 oxygen isotope. So you can write a very short answer. I will move on to the next question. Additional question 10. An activity to show oxygen is produced during photosynthesis is shown in the diagram below. Label the parts of the diagram. So you know this activity, you have observed it being carried out in the lab and you have answered questions related to this. Here you have to label the parts of the setup. What is A? The gas, that is gas collector. What is B? We have here, it is an aquatic plant. Here students, by looking at the diagram, we can't say whether it's hydrilla or vallisneria. So it's always better to write it as aquatic plant. And if they ask you for examples, then you can write the name of the plants. Then C is water. And D, here you can see, is the trough. And E is the glass funnel. Glass funnel. What is the method of collection of oxygen in the above setup? You all know oxygen is lighter than water, so it goes up and the water is pushed down, or water is displaced downwards. That is the downward displacement of water. So here you have to write that downward displacement of water. So that is the method of collecting oxygen gas. The next question, how will you make sure that only oxygen is collected in the boiling tube? How did we make sure? By filling the test tube completely with water without allowing any air bubbles. By doing that, we made sure that only the gas, that is oxygen, evolved by the plants is collected in the test tube. So here you have to say, fill the boiling tube completely with water without trapping any gas bubbles inside. So by doing this, we can make sure that there is no air or gas within the test tube, but only the gas that is evolved by the plant will be collected within the boiling tube. How will you confirm that the gas collected is oxygen? By inserting a glowing splinter. By inserting a glowing splinter. What is the significance of oxygen in biosphere? Oxygen is needed for aerobic respiration and combustion. So, it is needed for the survival of organisms. So, here what is the significance of oxygen in biosphere? Then we don't need to write combustion. We only need to say about the aerobic respiration. It is necessary for aerobic respiration. Therefore, it is important for the survival of many living organisms. The organisms that need to respire oxygen. Those are the aerobic organisms. With these students, I have come to the end of additional questions. So, I am sure you all have a very good understanding of the process of photosynthesis. You know the factors that are needed, the product that is produced, the main product and the byproduct, and how the main product is converted. And you know to write the equations, as well as I hope you have understood all the activities related to this lesson. So with that, again listen to the discussion, read your textbook well, and then answer as many questions as possible. That will make you competent to answer any type of question related to this lesson.